Hello everyone, welcome to Biotechnica. So today I have come up with the most important topic that you have to study, or we can say it's very important for you during your CSIR NET Life Science preparation time. And since we are at the last minutes of preparation, so it's very important what you have to study during this period. So today I have come up with the most important or the interesting topics, which is going to be the 3 type of question that you must attend in your examination from unit number 8, which is going to be inheritance biology, or we can say genetics. So let's proceed on to the video. So what are the 3 type of questions that you have to attend from inheritance biology or genetics? So we can say the first important thing you have to note down is pedigree analysis. So make sure you're attending pedigree analysis during your examination. And pedigree analysis usually will be coupled with probability questions, and the next is going to be complementation questions, and the next is going to be Tetrad analysis question, and of course it's going to be lot score also will be coming. So you'll be seeing that in the next video, in the upcoming video. So first we'll talk about Tetrad analysis. So here I'm going to very specifically talk about ordered Tetrad analysis. Okay, so let's talk about what is ordered Tetrad. So here it usually takes place in fungus like Ascomycetes, like Neurospora fungus. So here you can see this is going to be the cell, or this cell which undergoes meiosis. So here the first meiotic division will take place, where you can see it is getting separated here, which means the cell is formed here in the first meiotic division itself, which means when there is no crossing over, this is a condition. If there is no crossing over, suppose let's consider this is from the father and this is from the mother. Usually what will happen, these two will undergo crossing over. But in this case, we are talking there is no crossing over. In such situation, what will happen? Without undergoing any crossing over, not like this, what will happen is, suppose let me make it like this. So this is going to be like this. And the mother is going to have a chromosome like this. So what will happen is, instead of undergoing crossing over, this will become one cell and this will become one cell. There is no crossing over here, which is the first meiosis. So here, very important thing, when there is no crossing over, the first division segregation will take place. Division segregation, this is an important point to be noted. Based on this, we are going to do some questions. So remember, when there is no crossing over between 2 chromosomes which comes from the parent in the zygote, or we can say they will never undergo any kind of crossing over. If no crossing over, no recombination. First division segregation will take place here. Then what will happen to the next situation? This will separate. So you're getting a haploid set. This is diploid. This is diploid, and here you're having a haploid set, haploid set, haploid set, and haploid set. So capital A here, capital A here, small a here, small a here. Again, what will happen? They will undergo mitosis. If they undergo, this is haploid. If they undergo meiosis, it will become N by 2. So it will undergo mitosis. So here you're seeing 4 haploid sets. 4 haploid sets. So we are calling it as what? Tetrad. But in this case, you're seeing capital A, which is multiplied 2 times. And here capital A, small a, small a, small a, small a. So you're forming 8 cells, which are going to be 8 haploid cells, which are going to be haploid in nature. So we can say it is going to be octet. So you should remember one important point: when there is no crossing over between the parent chromosome and or we can say the mother and the father's chromosomes, there's going to be first meiotic division will take place, where you're going to get 2 cells. Second case is when there is going to be a crossing over occurring. Just check over here. Let me take, this is going to be 1 chromosome, which is from the father. So I'm just considering this is going to be the father. And there's another chromosome which actually comes from the female, or we can say from the mother. So I'm just making like this. So what exactly happened is, suppose let me consider this is capital A, this is small a and small a. Suppose if the crossing over is taking place between these 2. So what are the probability it can have? The next stage you can see, you can have something like this and this. But in this case, it will become what? small a will come here, but capital A will be here. In this case, it's going to be like this. So here it is going to be capital A here, small a here. So here you are observing capital A, capital A, small a. So when this is going to divide haploid set, capital A separately, haploid set, small a separately, capital A separately, small a separately. So here you're going to see capital A, small a, capital A, small a, small a. If I have to make this 2, 2, 2 and 2. If you're going to see when there is a crossing over between these two things, you're going to see 2, 2 and 2 and 2, which means 8 haploid sets are formed which are arranged like 2, 2, 2 and 2. This is very important. This is one of the situation. So here when there is a crossing over, you can get an octet set which are haploid in nature in the form of 2, 2, 2 and 2. This you should remember. The next important thing, suppose if the crossing over is taking place between the first one and the third one. Suppose this area is capital A, small a, small a. If the crossing over is taking place between these two things, what are the possibility? Capital A, small a, definitely it's going to be. You will have something like this. So you will have something like this. Here you will get small a. Here you will have small a. And this case you will have capital A here. And let me make this green color. Let me make this complete one as green color. This is completely green. So here you're having small a, capital A, and here you're having capital A, small a. So it has just changed. So what exactly happens now? It will become capital A, small a, small a, small a, small a, capital A, which means 2, 4, 2. So if you see an octet haploid cell which are in 2, 4, 2 or 2, 2, 2 and 2, then this is going to be when the crossing over is happening. This is second, we can say second division segregation. So remember, first division segregation, you can see the cells are here. Second division, it is happening like this. Small a, capital A, small a, capital A, small a. So remember, if there is a crossing over, it is going to be second division segregation, which can be, the cells can be 2, 4 or 2, 2, 2, 2 and 2. So I'm writing it as SDS. If the crossing over is not happening in the first meiotic division itself, there is a division. So it is FDS, which is going to be, we can say 4 capital A and 4 small a, 4 and 4. To summarize this one, let's write everything together. If crossing over is not there, no crossing over, then you should remember first meiotic division segregation will take place, which will be 4 and 4, which means capital A, capital A, capital A, capital A, capital A, capital A, all haploid cells. If the crossing over is occurring, there are two chances. So the arms can just interchange. Then it will be 2 and 4 and 2 or 2, 2, 2 and 2. This is second division segregation. Based on this only, we are going to do questions. Let's go for the question. Okay. The following table summarizes the result of cross between two strains of Neurospora. They have given 2 alleles. So the gene is going to be D gene, which has capital D and small d. Suppose let's write it like this, which is capital D here, small d here. The table shows different pattern of octet, which means they're talking about haploid set, which can be any way possible. And the number of ascus. Ascus is nothing but this one only. This is ascus sac, the complete one which is inside. And everything, we call it ascospore. Each individual are called as ascospore. So let's see. So here you're going to see this box, 4 capital D. Octet, this is one octet. So 8 cells, all are haploid. So first 4 are capital D. The next 4 are small d. So you're seeing 4 and 4, which means there is no crossing over. If there is no crossing over, they are first division segregation. Let's see the second column. Small d 4 are there and capital D 4 are there, which means they are going to be first division segregation. So the first two box, there is no crossing over. If no crossing over, you should remember the final ascospore sac is going to have 8 ascospores. All are going to be 4, 4 in number. The next column you can see, let me make it in a different one. Capital D 2, small d 2, capital D 2 and small d 2. Okay, so it is going to be 2, 2, which means this is SDS. The second one 2, 2, 2 and 2. So this is also SDS, which means crossing over is happening. This is also 2 and here there's going to be 2 and here there's going to be 2, 2, 2 and 2, or we can say 4, 2, 4, 2, SDS. So remember 2, 4, 2 or 2, 2, 2 if it comes SDS, which means crossing over is happening. If 4 and 4, it is first division segregation, which means no crossing over. This you should remember. So this is also SDS. This is also going to be SDS. So now I'm going to tell you the formula for how to calculate this one. So if you have to find out what is going to be the map units, you should remember the most important formula. So the formula is going to be the total number of ASCO, or we can say SDS. Tell you the formula for this one, how to calculate the map units is equal to half into the number of SDS, which means the second division segregation, divided by the total ASCUS, or we can say ASCII into 100. This is the formula. So we know this is one as SDS. I told you, which is in 2, 2, 2 and 2. This is also SDS. This is also SDS. And this is also SDS. So we have 14 and 16, which makes around 30. 30 and then here you're going to have 17, which is 47. And here you have 13, which is going to be 60. So if I have to make a formula for this one, half into 60. So 14 plus 16 plus 17 plus 13 divided by the total. The total you have to calculate everything. They have given 300. Divided by 300 into 100. That's all. So if you're going to do, it becomes 3 and 1 by 2. It is going to be 60 by 3. So it's going to be 20. So 2. Totally it's going to be 10. So the map units for ordered tetrad, here it is 10. So where are you seeing 10? You have to go in for the rejection method of option C. So let's check for the first one. The first two columns are from meiosis, correct? With no crossover. If you find no crossover, it is first division segregation, which happens in the first meiosis itself. The cell will divide. So here they have given between D and which of these things. Very important. If you have to find out, it is going to be the distance. Tetrad analysis is the distance between, we can say, the centromere and alleles or genes. This is important. So here they have given D and it's going to be centromere here. The distance between centromere and the allele is going to be, or we can say very specifically, suppose here there is capital D. What is going to be the distance between them? This is the centromere. So this is going to be this. So the first two columns are from meiosis, whereas no crossing over, FDS both are. And it is between allele and they have given centromere. So your answer will be in this because 10 is seen here. But let's check whether it is first division or second division. The pattern of first two column are definitely first FDS. So first division. And the map unit we found out 10. So the answer is very simple. It's going to be option B. Let's do for the second question. In Neurospora, a cross between genotype A and A is there. You can see first check in. This is a octet tetrad. So you have to see how many haploid cells. There are 8 haploid cells. All are haploid. And this is 2 of them are there. 4 of them are small a, and small a, 4 and 2. If you're going to see this pattern, you should remember it is SDS, which means second meiotic division when the cell will divide and there is crossing over taking place. Okay, now I told you how you can find out the distance. So the crossing over usually takes place between the centromere and the allele. Centromere and the allele. So the crossing over, here we are talking about crossing over. So in the centromere and the allele only it will take place. So we can say the first option is correct. Let's check for the second option. A and A in meiosis 1 they have given. No. Whenever crossing over happens, it is second division. So this is wrong. So it is always going to be meiosis 2. A and A in meiosis 2, correct. So it has happened like this. In meiosis 2 only, the division would have happened here, capital A, small a and small a. So from here only, the division would have happened. So the third option is correct. So these two are correct. So where are we seeing it? A followed by C. So the answer is very simple. It is option A. Okay, let's go for this third question. Very easy question. Find the distance between gene, or we can say allele, and centromere. Okay, so here they are talking about two allele, which is capital B and small b. How to calculate this is part C question. Calculate the distance between the gene and the centromere. We already know the formula. If you have to calculate the distance between the gene and the centromere, half into number of SDS, which means crossing over how many has formed, and the total ascus into 100. So it is going to be half. So let's check how many number of SDS. So how to find out? Just go for capital B. So it will be capital B, capital B. And the second one, capital B, capital B. I'm writing 8 haploid sets. So 1, 2, 3, 4, 5, 6, 7, 8. Just duplicating it. Here, small b. And for this one, small b, capital B, capital B, capital B, capital B. So 8 haploid sets I'm making it. Capital B, capital B. First one I'm taking. And the second one, small b, capital B, capital B, small b, small b. The third one, I'm taking first one, small b, and the second one, capital B, small b, capital B, capital B. The fourth one, first I'm taking capital B, small b and small b, small b and small b, capital B and capital B. And here small b, capital B and capital B, capital B and capital B, small b and small b. So let's now check. First, you have to remember two important things as I already told you. You should remember. First thing is going to be SDS 242, 2222. This you should remember, which means crossing over is happening. If no crossing over, first division segregation, which will be 4 and 4. 4 all will be similar, 4 all will be similar. So let's check 2 and 2 and 2 and 2. So these 4 are similar. So 4 and small b and small b. So this is going to be first division segregation. Let's check. This is also 4 small b, 4 capital B. So this is going to be FDS. And here 2 are capital B, 2 are small b, 2 are capital B and 2 are small b, which means 2, 2, 2 and 2, which is going to be SDS. And the next one, 2 are small, 2 are capital, 2 are small and 2 are capital. So 2, 2, 2, which is going to be SDS. And the next one, 2 are going to be capital and the 4 are going to be small. 2, 4 and 2, which is also going to be SDS. The next one is also going to be 2, 4 and 2, which is also going to be SDS. Now what we have to do, wherever you're seeing SDS, you're going to add. So here 4, 6, 3 and 7. So it is 10 and 10. So number of SDS is going to be 10. So half into 10, total ascus you have to calculate 36, 44, 4, 6 is given. So it is 20. So here you're seeing 30, 40. So totally is going to be 100. Sometimes they can give you the total values or you have to calculate 100 into 100. Just cancel this one and it becomes, or we can say this is going to be 20. Yeah, this is going to be 20. This is not 10. This is going to be 20. So it becomes 10 here. So the answer is very simple. If they ask you the distance between the gene and the centromere, half into number of SDS, you have to calculate SDS means 2222 or 242. And the answer is very simple. It's option 10. Okay. The next is complementation question. Very important question. Let me talk about what is complementation. Complementary means both go hand in hand together. Suppose I'm giving you one example. Suppose there is going to be one gene, which is gene A. And there's another gene, which is gene B. If both are present, then only there's going to be some reaction will take place. Suppose let's consider there is a flower, which is white in color. And whenever a gene A is expressed, they are going to produce one enzyme. And this enzyme is going to give some white color. Let me make it like this. And Gene B is going to produce an enzyme which is 2. And this white color changes into purple color. Suppose let's take a situation. If this gene A is not present, it can be the precursor, let's keep it. It's a precursor. Let's not put white color, we'll make it as a precursor. If precursor is not going to have this enzyme, will there be production of white color? No. If there is no production of white color, will there be production of purple? No. Let's take the second situation. Gene B is not present. Gene B is not present. So precursor is there and gene A is there. So enzyme 1 is there. So you get only white color. So if you want a purple color, both the genes should go hand in hand. So that is going to be complementary. But here we are going to talk about complementation test. When do we use this complementation test? This complementation test, we use it for heterozygous alleles or mutant ones, we can say, condition. So let us take some examples. Let me talk about a Drosophila. I'm taking one Drosophila which is going to be mutant, not wild type. There's a mutant male and a mutant female. So what is going to happen if the male is going to have one recessive allele, which means capital A, small a is there. Because of that, they are going to have white eye. If one recessive is there, then they are going to express white eye. The female is going to have, suppose one gene is mutated, so small a, capital B. So because of this recessive gene, it is also going to be white eye. So if they are going to undergo crossing over, or we can say or mating, the offsprings which we can see here is going to have capital A, capital B and small b, which means they have recessive from both the cases, but the dominant genes are present. So now they will not give any white eye, but they have red eye. We already know this. So what exactly happens? If both of them, both the genes are going to be present here, they are expressing what? Red eye. If any one of them is not present, they would have expressed what? White color, which means complementation will happen together. Very simple. Wherever you see positive, means there is a formation of genes like this. Capital A, capital B and small b. Wherever in question paper you see negative, which means complementation is happening. If it is positive, happens. If you're seeing negative, no complementation, which means two genes are not giving complementary to each other. This you should remember. Positive means complementation, negative means no complementation. So let's see one question which is given. A 7 arginine of E. coli were independently isolated and they have given positive. Just check. Let's consider these are all going to be genes. So complementing, usually you can calculate it for genes which are present on the same locus or on the different locus. So here, this is one gene and one gene. Let's take. So gene 1 and gene 2, you're seeing if they are getting crossed, what exactly happens? Same gene. So they are telling negative, which means no complementation. So which means gene 1 is in one group. So this is gene 1. Let me make all the genes. And when 1 and 2 are getting crossed, you're getting positive. So 1 and 2 will not be present on the same location, will be on the different. So 1 gene will be here and 2 gene will be here. Then only you have crossing over, right? That's what we are talking. So 1 is in one group and 2 is in one group. That's why there is going to be complementation. Let's talk about 1 with 4, 1 with 3. Again positive. So 3 is in a different group. So I'm writing 2 and 3. So 1 is in one group, 3 is in one group. And if you're going to see 1 and 4 is positive, so 1 is in one group, so you have to put 4 in different group. And in 5, again different group, 1 and 5 are in different group. And 1 and 6 are showing negative. 1 and 6 are showing negative, which means both are present in the same group, 1 and 6. The next one, 1 and 7 are also showing negative, which means 1, 6 and 7 are in the same group, 2, 3, 4, 5 are in the different group. Let's check for the second one. Second, 2 and 2 negative, 2 and 3 positive, which means 2 and 3 are in different group. But I've written 2 and 3 in, so I'm going to make this is a different group. Let me write it like this. And 2 and 4, definitely 2 and 4, this is one group. So let's take this is one group, this is one group, this is one group. So 2 and 4 are in different group. Yes. And 2 and 5, same. So 2 and 5, we can say negative. Yes. The next one, 2 and 6 positive, 2 and 6, so I'm not writing 5 here, I'm writing 5 here. So it has to be like this. And the next one, 2 and 6 positive, so 2 is in one group, 6 is in other group, so the complementation is possible. The next one is positive. After this, when you're going to see, right, the same thing will happen. So 1, 6, 7 are in one group, 5 and 2 are in next group, 3, 4 are in third group. So how many complementation you're going to see? 3 complementation, and there are going to be 3 genes. So complementation, you should remember, they ask you what is the complementation here. So there are 3 complementation groups. This is what you have to remember for the exam. Okay. So you can just take a screenshot of this one, this kind of question usually you will get. Just go positive means complementation is occurring and negative means there is no complementation. Based on this you can analyze. You can also pause the video and solve this question. The next is probability questions. So probability questions you have to remember 2 important things. One is going to be addition rule and multiplicative rule. Suppose if I have to talk about multiplicative rule. And then addition rule, additive rule, or we can say addition rule. In multiplicative rule, you have to remember, let me take one example. There is going to be 2 person, person number 1 and person number 2. This person is going to play football. And this person is going to play, let's take hockey. So these are 2 individual events, or to be very specific, there is going to be head and a tail. Let's take a coin. We can say a coin has 2 sides, which is head and tail. What is the probability of getting head? Definitely 2. In that only one time you can have head. What is the probability of getting tail? Only one time you can get tail. But in multiplication rule, if I have to talk about, both are individual events. When you get a head, it is different. When you get a tail, it is a different event. But we will talk together. What is the probability of getting head and tail together? So how will I do it? 1 by 2 into 1 by 2. So your answer comes around 1 by 4. So you have to multiply but together. In additive rule, it's just the opposite. Here what we will do is, we will just add, but remember both are going to be either way, mutually exclusive. So remember this one. So there is a person 1 differently, person 2 differently, which means they are playing football, another one is playing hockey. But I'm going to talk about both of them together. What is the probability of the person plays football and the hockey together? That is multiplicative rule. In additive rule, there is one person. This person can play both, either football, I'm telling or he can play hockey, or rule. This is going to be or. So additive rule is called as or rule. He can either play a hockey or he can either play a football. In additive case, you have to remember it has to be addition. 1 plus 2 plus 1 plus 2, which becomes 2 by 4, which has to be 1 by 2. So remember, multiplicative rule are going to be 2 individual events. But if I have to talk about both the events together, I'm going to multiply 1 by 2 into 1 by 2. But in additive, you're going to add, but they are going to be same event, but a person can either like that or this, or events we can say. Let's solve one question, very easy question. So here you're seeing a pedigree, inheritance pattern. So this is going to be, they have given recessive disorder. Let's understand pedigree. First important thing you have to understand in a pedigree is whether the affected individual has affected parent. Let's check, this is the affected individual. So he don't have affected parent. This is the affected individual. He don't have affected parent. If you get yes, then it is going to be dominant. Affected individual, affected parent. If your answer is going to be yes, then it is dominant. But here we don't have that. We have no for this question. So it is going to be recessive. So we come to a conclusion, yet yes, it is recessive. Second question you have to ask whether it is going from the son to, or we can say father to daughter to grandson, or from a mother to son to a granddaughter. If you see this, then you remember it is sex linked. So it is not going like this. So it is going to be definitely autosomal recessive ones. So let's consider Recessive ones means if small a and small a is there, then it is diseased. So here it has to be capital A, small a, capital A, small a, capital A, small a, capital A, small a. If it is small a and small a, it is disease. So they have not shaded. Here they have shaded, which means small a and small a, small a and small a. The rest of the things are going to be capital A, small a, capital A, small a. The question is, a child from a marriage between individual 2, which means first generation, second generation. This is the individual 2 and the individual 3 are married. They show this disorder only if the parents carry the recessive gene. Yes, they both are carrying one of the recessive genes. Small a, they are carrying it. What is the probability that the child will show the disorder? Yes, they didn't say how many children. They are asking the total probability. So what we have to do? We have to total together means what you have to do? Multiplicative rule. So which means we are going to do some multiplication here. First, let's check this part. Capital A, small a, capital A, small a. If you're going to do this, what you'll get? Capital A, capital A, capital A, small a, capital A, and small a, small a. Out of these 4 individuals, or let me write it here, capital A, capital A, capital A, small a, capital A, small a, small a, small a. Out of these 4 individuals, only one is disease. So what is the probability? 1 by 4. But do not come to a conclusion. The answer is going to be 1 by 4. They ask the total together. You have to check how many are going to be without deceased. So if you see how many are without disease, 3. So out of that, how many is going to have recessive? 2 by 3. So there is a probability that they can be 2 by 3 into 2 by 3 into 1 by 4. You have to remember this. In order to make it very simplified, we can say, so capital A, or we can say the father is going to be, this father is capital A, small a, capital A, small a. So what is happening? There's going to be capital A, capital A, capital A, small a, capital A, small a, and small a, and small a. So out of these 4 individual, only one is diseased. So I'm writing it 1 by 4 is deceased individual. The remaining are going to be normal, but out of these normal are going to be 3. Out of these normal, how many are carrying the recessive genes? 2. So the probability is going to be 2 by 3. So there is a probability 2 by 3 chance is also there. 2 by 3 chance is also there, and 1 by 4 deceased is also there. So how can I write? 2 by 3 into 1 by 4 into 2 by 3. So what should be the answer for us? It will be 4 and it will be 12. So it becomes 1 by 9. See, so 1 by 9 you're seeing only in first option, 1 by 9, and the probability of the parents carrying the recessive gene is 2 by 3 probability. The parents, if you're talking about this, 2 by 3 probability they can carry. 2 by 3 probability the parent can carry, and the child will be 1 by 4. That's why we are multiplying 2 by 3 from the parent, 2 by 3 from the parent, and 1 by 4 the offsprings are going to be affected. So totally it's going to be option 1. Let's do the last question and we'll finish off. Very easy question. The following pedigree shows the inheritance pattern of a trait. Yes. So they said select the possible mode of inheritance and the probability that the daughter in generation 3, this is 3, this is 2, this is 1, will show that. So we have to check whether it is X-linked or Y-linked or everything. First check for Y-linked. Y-linked means all males affected. All males affected. Let's check. Here the male is affected. Here the male is affected. But here the female is affected. Here the male is affected. So it is not a Y-linked. So just going for rejection method, Y-linked. Let's check X-linked. X-linked, I already told you, if it's going to be a X-linked, it goes from male to female to male, or from female to male to female. So check, from male it is going to male, and from female it is going to male. So you cannot apply this, so it is not sex linked. So we will cross first option, second option. Let's check for the third and the fourth option. Autosomal recessive, definitely we know. So let's write, this is capital A, small a, this is disease, small a, small a, small a, small a, capital A, small a, small a, small a, capital A, small a, capital A, small a, and small a, small a. Now we have to find out the third generation only. Let's do capital A, small a is the female, and the small a is the male, which is affected. If you have to do a cross, what you'll get? Capital A, small a, and you will have capital A, small a, small a, small a, small a, small a. Out of 4 individual, 2 individuals are affected. The question is the third generation will show this trait. So 2 are affected and the total offspring is 4. So it is going to be 1 by 2. So what is going to be the answer? Very simple, autosomal recessive. Probability is 1 by 2. The next is a very easy question. You can see, go in for this option. First you have to see whether it is Y-linked. Y-linked means only males are affected. No, females are affected here. So it is not Y-linked. X-linked, male to female and female, but it is happening everywhere. It is also not X-linked. So you have to reject first option, second option. Then the remaining is going to be only C and D. So the option you can see only in second, which means mitochondria. Mitochondria means it usually goes from mother, the disease goes from the mother to all the young individual, all offsprings. So if you know option C, then you can go for the second option. It is variable according to the heteroplasty, different situations. So the answer is very simple. It's option C. So if we have to talk about inheritance biology, the most important thing is probability questions, pedigree along with probability questions, we can say. Lot score is another important thing which we'll be seeing in the next videos. And the second important thing, tetrad analysis based on first division segregations and second division segregation. And the third important thing we already talked about, complementation test, which is also going to be very important. So if any 3 type of questions, if it comes in your genetics chapter, never miss. It's going to be very simple and try to solve a lot of questions. And thank you all of you for your time. And I'm going to meet you in the next video. Thank you so much for joining. Yes.